Imagine a world where the very air we breathe, the water we drink, and the food we eat are all contaminated with tiny plastic particles. It's a daunting reality that scientists are working tirelessly to understand, and it all starts with detecting and measuring the amount of plastic particles in human bodies. But here's the shocking truth. While it's relatively easy to pull plastic particles out of seawater, finding and tracking them in human body tissue is a far more complex and challenging task. A new expert review has shed light on the methods labs can use to detect plastic particles in blood, lungs, placentas, and other tissues, and why the choices they make will shape what health risks they reveal. At the heart of this issue is a simple yet daunting question. How do you measure plastic inside living systems without distorting the evidence? According to Baoshan Xing, professor of environmental and soil chemistry at the University of Massachusetts and lead study author, every biological sample represents a different matrix, which drives the lab playbook. This means that fatty tissues, protein-rich organs, shells, and plant fibers all require tailored digestion steps to separate plastics without melting or morphing them. But what exactly are microplastics and nanoplastics? Microplastics refer to solid plastic particles roughly 1 micrometer to 5 millimeters in size, while nanoplastics are particles smaller than 1 micrometer, often defined by colloidal behavior in the 1 to 1,000 nanometer range. So, how do researchers find plastics in the lab? After digestion, labs isolate particles and verify their identity using tools like Raman spectroscopy, FTIR spectroscopy, and thermal methods like pyrolysis gas chromatography mass spectrometry, GCMS. Each of these methods comes with trade-offs in size limits, throughput, and polymer specificity, especially in complex tissues. And this is the part most people miss. Quality control matters just as much as the chemistry, including strict contamination blanks, clean air handling, and reporting of detection limits to prevent false positives or inflated counts. But what do early health signals show? Researchers have measured microplastics in human blood, showing that polymer fragments can circulate systemically. They've also detected measurable mass concentrations using pyrolysis GCMS. Follow-up work has begun to profile sizes, shapes, and polymer types in blood from healthy volunteers to probe possible links with physiology. And here's where it gets controversial. Scientists have found plastic particles in human placentas on both the mother's and baby's sides, raising concerns about exposure before birth. Human lung tissue examined after surgery has also contained identifiable microplastics, and vascular studies have reported microplastics and nanoplastics in carotid artery plaques, associated with higher risks of heart attack, stroke, or death over follow-up periods in clinical cohorts. But the methods used to measure plastic particles can alter the outcomes, and this is where the debate begins. Dose matters, but researchers can count it in several ways, including the number of particles per gram, mass per gram, or surface area per gram. Interpretation hinges on the metric, and different shapes behave differently, so researchers must choose measurement methods that fit the health question. As Singh noted, there are no accepted protocols yet, making it hard to compare plastic levels across studies or turn them into health guidelines. However, AI is helping to track plastics by pairing Raman datasets with classification models to speed up polymer identification and reduce analyst bias. Thermal analysis coupled with FTIR is also being automated with ML to tease apart polymer signals in complex biological matrices, improving throughput while keeping false identifications in check. So, what's the solution? Plastics research needs standards, including reference materials, interlaboratory comparisons, and matrix-specific digestion and detection protocols. Agencies are already coordinating methods of research to support that standardization push. As Xing said, this is a headache, but the upside is that a clear, shared toolkit would let scientists quantify risks with the same confidence we expect in other areas of environmental health. The study is published in Nature Reviews Bioengineering, And it's a call to action for researchers, policymakers, and individuals to work together to address the growing concern of plastic pollution in our bodies. But here's the question. What do you think is the most pressing issue related to plastic pollution in human bodies? And how do you think we can work together to address it? Share your thoughts in the comments below.